If you look deep into a human cell, past the biology, you find a sequence that looks less like a living thing and more like a massive, scrolling wall of raw text, just A, T, C, and G. This document contains over 3 billion individual base pairs. If you tried to read it like a book, you'd be turning pages for decades. But occasionally, a single character gets swapped. An A becomes a T. Biologists call these single nucleotide polymorphisms, and that one-letter variation can be the root cause of a hereditary disease. Identifying that swap isn't a lab experiment anymore. It's a retrieval operation across a string 3 billion characters deep. Traditional relational databases aren't built for this. If you try to process a full genome using standard software, the system stalls. These systems require rigid rows and columns, but you can't chop a continuous 3 billion character sequence into a table without losing the context of the genetic code. Standard indexing tools, like B-trees, were designed to organize discrete names and numbers. They aren't equipped to navigate unstructured, continuous text. Sequencing machines now produce petabytes of data. A simple linear search, reading the code from start to finish, would take a human lifetime to complete on a single machine. Standard transactional logic is useless here. The biotech industry had to build systems from the ground up that treat DNA as a searchable landscape rather than a ledger. The solution is genome data management, specialized platforms engineered to handle extreme algorithmic complexity and unstructured sequence strings. To solve a text parsing problem of this scale, computer scientists rely on a specific data structure called a suffix tree. The strength of this system isn't just holding petabytes of storage, but how it mathematically prepares the text before the search begins. In this animated diagram, we have a short sequence, ATCG. Instead of reading it linearly, a database prepares the text by breaking it down into every possible suffix or ending. Then, the algorithm uses these endings to build a branching tree. Suffixes that start with the same letters are grouped together, sharing a parent node. This branching topology allows the system to bypass billions of irrelevant letters during a search. Now, searching for a mutation takes milliseconds. The algorithm follows the illuminated branches to a precise coordinate tag, pointing to the exact location of the typo in the original sequence. The result is a structure that turns a billion-letter wall of text into a mathematical index where any sequence is reachable almost instantly. With the specific mutation extracted, the focus shifts to finding its clinical meaning. This is where the knowledge discovery in databases process begins. The patient's raw sequence data is integrated and transformed into actionable insights. The database cross-references the patient's specific mutation against massive global repositories of clinical oncology data. A specific mutation is just a coordinate until the system connects it to historical medical outcomes. Unsupervised clustering algorithms group the patient's specific tumor profile with the treatment records of thousands of other patients with similar mutations. This allows doctors to bypass generic treatments and prescribe therapies validated by data from the exact same string match. Treating genetic disease has moved from the petri dish to the database. It turns out, a more efficient way to index a string leads to a more precise way to treat a patient.